Where we left off last time is we had drawn the idea of the membrane-bound compartment called the endoplasmic reticulum, and we had decorated that membrane-bound compartment with ribosomes, and we had said that there are a subset of proteins which will be generated on those ribosomes attached to the endoplasmic reticulum, and we'll begin there today. We'll draw a kind of box that's meant to represent the endoplasmic reticulum bag. We'll then decorate that bag with ribosomes and describe that as rough endoplasmic reticulum. The distinction between rough endoplasmic reticulum and smooth endoplasmic reticulum is simply that rough endoplasmic reticulum is decorated with ribosomes and smooth endoplasmic reticulum lacks those ribosomes. But this is just the first compartment we're going to talk about. And we're going to begin, as we left off last time, with the decoration of those proteins in the endoplasmic reticulum with covalently linked sugars, so that inside the endoplasmic reticulum will be enzymes that attach sugars to those proteins that are being synthesized there. But the story doesn't end there. That in fact what we'll do is we'll start in the endoplasmic reticulum, we'll begin to decorate those proteins with sugars, but then we'll further process and manipulate those proteins in a new structure which is called the Golgi apparatus. So now we have to imagine how we're going to deliver proteins from the membrane-bound bag of the endoplasmic reticulum to the next series of membrane-bound bags called the Golgi apparatus. Functionally, what the Golgi apparatus does is it serves as a kind of factory where proteins are finally changed to their final structure and shape. So you can imagine the factory as a place where we hammer on these proteins to get them to just the right shape and organization and add some more sugars. But all three types of proteins that we mentioned last time that are first generated in the endoplasmic reticulum, that is the proteins destined for secretion, the proteins destined to become transmembrane proteins, and proteins destined to go to the lysosome, the so-called lysosomal proteins, those are then sorted here in the Golgi apparatus. The proteins that are destined to go to the plasma membrane and leave the cell by secretion, by exocytosis, those proteins are trafficked into a subset of vesicles. Those vesicles now are then going to be targeted to the plasma membrane and they'll go to the outside world. But other vesicles are going to be loaded with lysosomal enzymes. Those lysosomal enzymes, and these lysosomal enzymes are pretty caustic. I mean that in terms of being proteolytic enzymes. They're enzymes that break down other molecules. They digest proteins. They digest lipids. So we're going to have vesicles that contain these proteins that are pretty destructive. So what we'll try to do is sequester these proteins, these destructive proteins, inside a membrane-bound compartment, which will be called the lysosome. All of this is going to be talking about how we package proteins into membrane-bound compartments called vesicles, these cargo-carrying containers, and then guide those cargo-carrying containers either to the plasma membrane or to the lysosome to deliver the appropriate proteins to the right spot. And we'll talk a little bit about how that gets done. But just as much of this is going to hinge on the idea of exocytosis, the fusion of a vesicle with a plasma membrane to dump things to the outside world, there's also going to be a process called endocytosis, where we're going to bring things in from the outside world into the cell. And one of the places where we want to do that, one of the examples of where we want to do that, is we're going to use the example of endocytosis in a subset of cells called the macrophages. The macrophages are an important part of your immune system. We'll run into these later in the course. But for now, I want to simply point out that a macrophage is a specialized cell, and that the meaning here, macrophage, is to be the big eaters. And their process of eating is, in fact, endocytosis. So what they're going to do is they're going to bind to pathologic bacteria on the outside of the cell, bring those pathologic bacteria into the cell, keep them encased within a membrane-bound compartment we call a vesicle, and guide those vesicles now to the lysosomes, where we can now destroy that potentially pathogenic bacterium in the lysosomes. So we'll talk a little bit about how that gets done. And then we want to talk about this last of the membrane-bound compartments. The last of the membrane-bound compartments is the mitochondrion. The plural is mitochondria. The mitochondrion plays a really, really important function for us, because what the mitochondrion does is it uses oxygen in a process called oxidative phosphorylation. It uses that oxygen through a series of oxidation and reduction pathways to use the oxygen to make something known as adenosine triphosphate ATP. Almost all of your enzymatic activity inside the cell is driven. Those enzymatic activities which require energy, almost all of them are driven by adenosine triphosphate. You can make adenosine triphosphate with enzymes that are in the cytoplasm, but not much gets made that way. The lion's share of the adenosine triphosphate inside all of your cells is produced by something called oxidative phosphorylation, which requires oxygen from the air, which requires the redox reactions where you now hand oxygen off to a cytochrome and generate ATP at each of those steps. Now, we're not going to go into the biochemistry of that whole pathway. But it's a wonderful story as about where these mitochondria came from. They have a great history, both scientifically and biologically. And it looks like the mitochondria that we have inside our cells are all related to a single capture event that occurred probably 1.5 billion or more years ago. That some long ago single-celled primitive eukaryotic cell captured one of these mitochondria. But at that time, they weren't called mitochondria. Well, they weren't called anything, but they weren't mitochondria. They were probably bacteria of some sort. So there was a capture event of this bacterium, but this bacterium had learned to live in a new environment where there was lots of oxygen, and it was able to use that oxygen to make ATP. And so what we think happened is that some predatory cell gobbled up one of these bacteria that had figured out a way to use the oxygen-rich environment to make ATP, and that bacterium set up housekeeping inside that cell. And it looks like all of the eukaryotic cells from that time forward, let me put it this way, that all of the eukaryotic cells in your body are evolutionarily related to that single capture event. All of the eukaryotic cells you see in the world around you are probably related to that single capture event. So there was this peculiar case long ago where we captured a bacterium and have since that time lived symbiotically with that bacterium to make ATP. And I'll tell you a little more about that story. Now, if we ended at this point, you would be forgiven of thinking about the organelles, the membrane-bound compartments inside the cell, as being vegetables in minestrone soup. But they're not. They're highly organized. It is not a random structure inside the cell. The cell is highly organized by a structure called the cytoskeleton. So we'll spend a little bit of time, probably tomorrow, on the structure of the cytoskeleton. The cytoskeleton that we're going to talk about is composed of microtubules and actin. One of the hallmarks of these proteins is that they are polymers of proteins. They are proteins, gigantic proteins, put together from subunits. In the case of microtubules, they're put together from subunits of tubulin. Filamentous actin is put together from subunits of actin. But it's not just that we make gigantic proteins out of other proteins. It turns out that both the microtubules and the actin have a kind of endedness to them, a kind of plus end and a minus end. They have an orientation. And that orientation imparts function, because that orientation allows for motors to attach to microtubules or actin. One set of motors called kinesin will now move only in one direction along the microtubules. These are said to be plus end-directed motors. Dynein is a minus end-directed motor, so it moves in the opposite direction along microtubules. And myosin, too, is a motor, but now it's restricted to moving along actin. But now what we have is a series of rails. Whether those rails are made of microtubules or whether those rails are made of actin, we have rails along which we can move cargo-carrying containers with molecular motors. And we have a directionality on those rails depending upon the motors we use. And we'll talk about that in some detail.